Hi and hello everyone. This is Dr. Kalpana V.N., the Department of Microbiology from Antigen College. Well, I'm here to explain about the basis of antigen specificity. What do you mean by specificity? It is nothing but an ability to recognize and respond to one particular antigen among a different antigen. That is called specificity. For example, when a particular antigen X, when it combines with a B cell receptor of B cell, the B cell will get activated. So the plasma cell, which has been derived from this activated B cell, gets activated by the antigen X that will produce an antibody against X. That antibody we call as anti-X antibody. Now this anti-X antibody will combine only with the antigen X but not with any other antigen. So the antibodies produced against an antigen will react specifically with that antigen only. There are numerous microorganisms and other potentially dangerous antigens present in the environment. Whenever a human is exposed to these antigens, they may enter into the body and cause damage. So we already know that the immune response induced against one antigen is specific to that antigen only, and these immune response will not act against other antigens. That means there should be innumerable number of T cells and B cells and each capable of acting against different antigen. In fact, one of the remarkable features of the immune system is that it can respond to vast array of foreign antigen. To meet this demand, the host produces enormous number of lymphocytes, each having a different type of antigen receptor. So, the host immune system is already ready with arsenals which can tackle any type of antigen even before the entry of antigen. So during development of lymphocytes in the bone marrow and thymus, rearrangement of genes which code the antigen receptors occur. Because of genetic rearrangement, different gene sequences are formed. Consequently, the antigen receptors coded by the different genes will have a different antigen specificity. So we have some salient features of antigen that is like specificity, immune complex, binding site of antigen, and binding force of antigen. If you go with the antigen specificity, it mainly depends on specific active site on the antigenic molecule, that is antigenic determinant. Antigenic determinant or epitope or the regions of antigen which specifically binds with antibody molecule. You can see here this specificity refers to ability of an individual antibody combining site to react with only one antigenic determinant. So each antibody will bind to a specific antigen, an interaction which is similar to that of lock and key. So here you can find an immune complex. So integral binding of an antibody to a soluble antigen. So as a result, we have an antigen antibody complex. We have a binding site of antigen. The part of antigen which combines with antibody is called epitope, that is being recognized by immune system, specifically by antibodies like B cells and T cells. So the part of antibody which recognizes an epitope is called as a paratope. The binding force of antigen. The binding force between antigen and antibody in antigen-antibody reaction is because of three factors. One is a closeness between antigen and antibody, that is more close, good strength of binding. Second, it's a non-covalent bond or intermolecular force or hydrogen bonds, van der Waals force, hydrophobic bonds. And third is because of the affinity of antibody, that is the strength of reaction between a single epitope and a single paratope. Thank you everyone.